Hello everyone, my name is Christina Besada and I am a third-year graduate student at Drexel University College of Medicine in the lab of Dr. Ola Mortensen, and today I'm thrilled to be talking to you guys about my work in the lab, specifically on MKP3 and how it regulates dopamine transporter trafficking and function. Before we get into the crux of the research, I want to provide you with some brief pertinent background to our studies. To start us off, dopamine is a neurotransmitter produced in the ventral tegmental area, or VTA, and plays a critical role in motivation and reward. The VTA sends dopaminergic inputs to the nucleus accumbens, or NAC, via connection of the mesolimbic reward pathway. Tyrosine hydroxylase, or TH, is the rate-limiting enzyme in the dopamine synthesis pathway. Dopamine neurotransmission is regulated by the dopamine transporter, or DAT, and psychostimulants such as cocaine block DAT, preventing dopamine from being reuptaken back into the presynaptic neuron. Furthermore, DAT is acutely regulated by the protein kinase PKC, which is involved in the activation of ERK. ERK is a MAPKs that affects the function and expression of proteins that regulate dopaminergic tone and gene expression levels. Activation of this signaling cascade has been implicated in DAT phosphorylation, specifically on the threonine 53 site. Furthermore, the MAPKs phosphatase and ERK inhibitor MKP3, studied by Dr. Ola Mortensen and his colleagues back in 2008, was found to prevent the PMA-induced downregulation of DAT in vitro. So here's a quick diagram explaining their findings of their in vitro studies. So here are dopamine transporters on the cell surface. When PMA is introduced, it activates PKC, which results in dopamine transporter internalization. Dopamine transporter can then either go through degradation or get recycled back to the cell surface. When MKP3 is introduced, it inhibits this PKC-induced internalization, and thus there is more dopamine transporter expression on the cell surface. And since we know that PKC and ERK play a critical role in DAT regulation, and MKP3 is an ERK inhibitor, we wanted to further explore the effects it has on gene expression levels and DAT trafficking in vivo. In order to do that, we employed an in vivo model of TH-Cree rats. All rats utilized for our studies undergo a stereotactic surgery in which they receive a bilateral injection of our MKP3 viral construct into the VTA. We then perform a procedure known as brain slice biotinylation, and additionally we employed RT-qPCR tests in order to quantify levels of pertinent genes. To start us off, we did RT-qPCR to look at RNA levels of DAT, TH, and TBP. We expressed the final values as full change in gene expression compared to rats that received only GFP during their surgery. As we can see in both the males and females, there's a significant decrease in both DAT and TH RNA expression levels when MKP3 is overexpressed. We now want to see the effect of MKP3 overexpression on total DAT protein levels. The Western blot over here is showing us that there is more total DAT in the MKP3 overexpressing samples, and figure B is quantifying that for us. Next, we wanted to investigate the effect of MKP3 overexpression on DAT trafficking and the amount of cell surface DAT. Over here in figure C, we can see that with MKP3 overexpression, there is a significant increase in cell surface DAT. At first glance, this seems to contradict our RT-qPCR gene expression data. However, I will get more into an explanation later. If you remember, I prefaced earlier that Dr. Mortensen identified MKP3 as an inhibitor of PMA-induced internalization in vitro, so I wanted to show you some of that data. In figure A, we see a representative time course of MDCK cells expressing DAT or DAT with MKP3 after PMA exposure. On the top row, where the cells are just expressing DAT, we can see that during the 30 minutes, the cells start to have this punctated look, indicating that DAT on the surface is internalizing into the cell. However, with MKP3, we see that this effect is no longer there. Thus, MKP3 is preventing that PMA-induced internalization. The same effect was further confirmed in Western blot analysis after biotinylation. So here in figure B, we can see that there is reduced DAT cell surface expression upon PMA exposure, but when MKP3 is introduced, that effect is attenuated. We wanted to investigate if we could see that same effect of MKP3 preventing DAT internalization in vivo with our TH-CRE rats. We performed brain slice biotinylation with PMA exposure, and here we can see that with the wild type, there's a significant decrease in DAT cell surface expression. However, that effect is lost with MKP3 overexpression. Thus, this indicates that DAT in the presence of MKP3 is resistant to internalization. We now want to see the effect of MKP3 overexpression on the phosphorylation levels of DAT, because we believe this phosphorylation could be involved in DAT trafficking. I'm sure this graph looks familiar from earlier, so in addition to total DAT levels, we are also looking at the effect of MKP3 overexpression on the phosphorylated state of DAT. In this Western blot, we can see that there really isn't much of a difference of PDAT levels in our MKP3 and control samples, and this is quantified for us in Figure B. However, when looking at the ratio of PDAT to DAT, we can see that PDAT is significantly downregulated in the MKP3 group. We wanted to see if the same trend held true for samples exposed to PMA. As we can see here, the MKP3 group has less phospho-DAT levels in the presence of PMA compared to vehicle, indicating that MKP3 overexpression may inhibit phosphorylation of DAT. So just to recapitulate everything that was discussed here, over here is a little diagram tying together this information and summarizing what we think is going on. So on the left, where there is no MKP3 present, PMA is inducing internalization of DAT, where it can either get degraded or cycled back to the cell surface, and there is natural feedback occurring for gene expression of more DAT. However, when MKP3 is present, we are seeing a reduction in gene expression of DAT as indicated by the thinner arrow here. So, if there is less gene expression, how are we seeing more total DAT protein on the cell surface? Well, we know that MKP3 inhibits the DAT internalization, however, the mechanism is not completely known. And we think that phosphorylation is contributing to internalization. So, when MKP3 is present, it is causing a reduction in phosphorylation of DAT, resulting in inhibition of internalization of DAT. And we end up with an accumulation of DAT on the cell surface. This confirms that MKP3 overexpression is able to regulate DAT function and trafficking, and the lab is further exploring the mechanisms involved. And so, MKP3 has proven to be a very useful tool because we are just cracking the surface with what it's able to regulate in terms of DAT phosphorylation state, regulation, and trafficking. Previous data from the lab has even shown that MKP3 translates on a behavioral level because rats overexpressing MKP3 had less motivation to obtain cocaine. So our studies with this tool could really help to reveal novel therapeutic strategies for treating drug use disorders. Here are some future experiments that the lab plans on pursuing, and I would be happy to discuss them more in detail outside of the realm of this presentation. I want to thank you all so much for your time and for listening to this presentation, and especially a big thank you to both the Andrea and Ulla Mortensen labs for making this research possible.